Hi, it's Mr. Anderson, and in this podcast, I'm going to talk about
biotechnology. Biotechnology is arguably one of the most practical applications of bio, and it's also popped up on some previous tests, so it might be a good idea to have an understanding of what it is. So, what is biotech? And basically, it's humans playing God. More specifically, it's manipulating living things in order to make other products that people want. Although it doesn't seem like it, humans have actually been using biotechnology for almost our entire existence. Let's take these horses. Pardon the terrible drawing. This horse is strong, this horse is fast, so let's mix them together. Selective breeding is a simple example of biotechnology. But that's not the cool stuff. We want the Jurassic Park style test tubes and computers and all the gadgets and gizmos, and that's where we talk about genetic engineering, which is the main focus of biotech today. So let's get our feet wet, sink our teeth into the meat, and get right into it. In order to play with genes, you gotta have some first, and you can't just plop in one molecule of DNA. You're gonna need quite a substantial amount in order to do anything significant. So the first step is to clone your DNA. So let's say we have a diabetic over here, and you want to give his pancreas cells some insulin-producing genes. How do you do that? You have to clone the gene. And the oldest and probably easiest way to do that is to stick it inside a bacteria and let it do the cloning of the DNA for you. Now, as you've probably learned, bacteria can take in genetic material through horizontal gene transfer processes such as transformation, transduction, and conjugation. And the primary way they do this is with small circular non-chromosomal molecules of DNA that contain non-essential genes called plasmids. And these are the things that we can manipulate in order to clone DNA. We can stick the genes into the plasmids, stick the plasmids into the bacteria, and let the bacteria do all their meiosis stuff and make genes for us. Now, the exact process is a little complicated, but the main idea is that you have to cut up your DNA using restriction enzymes, which are naturally occurring bacterial enzymes that recognize specific sequences in order to protect the bacteria. So the restriction enzymes recognize restriction sites and create a bunch of restriction fragments, which are pieces of DNA after it's been cut up. Unfortunately, a DNA molecule may contain more than one restriction site, which means that even if you want to cut out a specific area with your restriction enzyme, indicated by the green on the picture, you may end up cutting out another restriction fragment that does not contain the desired gene. So now you can mix all your restriction fragments together with plasmids that have been cut up and hope that they bind together and form recombinant plasmids. However, because the restriction fragments may have been different, you can't tell which one of the plasmids actually contains the gene that you want. One of the ways that you can find out is by using a radioactively labeled piece of DNA or RNA that binds to the correct gene called a nucleic acid probe. Because of this, you can then culture your bacteria that contains your plasmids, cover it in the radioactive nucleic acid probe, and then check for radioactivity. The cultures that contain radioactivity thus contain the desired gene because the nucleic acid probe has bonded to the complementary base pairs. One downside of using bacteria is that they may not recognize some eukaryotic features of transcription and translation, such as intron splicing, which means that it may be more efficient to use eukaryotic cells such as yeast instead, which contain plasmids and thus are also compatible. Although using cells, either prokaryotic or eukaryotic, to clone DNA is pretty much the go-to way, an artificial method called polymerase chain reaction, or PCR, allows you to amplify a very small amount of DNA to a very large amount. The recipe calls for your DNA sample along with a special kind of DNA polymerase called Taq polymerase coming from thermophilic bacteria, as well as some special DNA primers. You then mix them into some hot water, and then voila, you get your DNA. There are some downsides, such as the DNA sequence must be pretty short, and you have to know part of the sequence in the first place in order to make your primers. However, PCR is pretty common for things such as crime scene investigations and testing for prenatal disorders. Now that you've cloned your DNA, you're ready to analyze it, and one popular way is using gel electrophoresis. In this process, you cut up your DNA with your handy restriction enzymes, dump it in some agarose gel, and turn on the positive and negative electrodes at each end. Since DNA has negatively charged phosphate groups, it starts moving towards the direction of the positive electrode. Smaller pieces are able to penetrate the gel further than large pieces, so they go further. So how does this actually help you find out anything? You're just seeing how far DNA actually goes or how small you made the piece. Well, if a mutation occurs in a restriction site, then the restriction enzyme can't cut the DNA in that area. You'll get a different number of restriction fragments than from another person who didn't have the mutation. This is called a restriction fragment length polymorphism, and this is one of the earliest methods of DNA analysis. For example, sickle cell disease can be characterized by a restriction fragment length polymorphism. And so when you test someone with sickle cell disease, their gel electrophoresis pattern will be different than from someone who doesn't have the mutation, since they'll have a different number of restriction fragments. So you can use this rule in order to determine whether someone has some sort of mutation that may be associated with a disease. Another thing you might want to do is find the sequence of your DNA. For example, if you're the Human Genome Project, that might be one of your prime interests. So the old method was to use the dideoxyribonucleotide chain termination method. The main idea is that you mix your template DNA with the proper enzymes and a supply of nucleotides. Then you add in some special fluorescent nucleotides called dideoxyribonucleotides, with one for each color, for each type of the bases, A, C, G, and T. When one of these gets added to a replicating strand, the replication stops, and it's completely random as to whenever one of these is added, so after a long time, you'll theoretically have at least one DNA strand up to any given point on the template strand. For example, if you have a 10 nucleotide strand, then you'd have a 1 nucleotide strand, a 2 nucleotide strand, and all the way up to 10. Then you could pass the DNA through a fluorescence detector in order to correspond the colors of the dideoxyribonucleotides with bases and therefore determine the sequence of the DNA. However, it should be noted that this practice was so last century, and nowadays we have machines that can tell the sequence of the DNA as it's being synthesized. Maybe you want to find how much a gene is actually expressed inside a cell. One popular way is called reverse transcriptase polymerase chain reaction. You take the mRNAs out of a cell, turn them into DNA using reverse transcriptase, which is an enzyme found in certain RNA viruses, and then use PCR in order to clone the gene. You can then use gel electrophoresis in order to measure the presence of certain DNA molecules. Another way is to inject some colorful nucleic acid probes and then measure the fluorescence to see how much of the mRNA is being expressed all over the body. Using this and computer programs, you can make DNA microassays that show the expression of hundreds or thousands of different genes together. What if you want to find out what a gene actually does? The best way to do that is to see what happens if the gene simply doesn't work. Using in vitro mutagenesis or RNA interference, scientists can disable certain genes and then observe their effects in order to determine the function of the gene. However, since we kinda can't disable genes in humans and watch what happens, what we can instead do is test for genetic markers that are associated with disorders. This is called a genome-wide association study. An example of these genetic markers can include single nucleotide polymorphisms, which is when there's a single base that differs in more than 1% of the population. Oh boy, we get into stem cells. There are three types that are going to be relevant to biotechnology, and that's totipotent, pluripotent, and adult stem cells. Totipotent stem cells can differentiate into all of the specialized cell types. Pluripotent stem cells can differentiate into many different types, but not as many as totipotent stem cells can. And adult stem cells can only differentiate into a few select specialized cell types. And the best types are, of course, the totipotent stem cells, because that's what we can use to make medicine, grow new limbs for people who've lost them, replace organs, do all the cool stuff. But we can only get those usually from embryonic stem cells, which means that we have to kill the embryo, and that's just not acceptable. So the solution that scientists are coming up with is that instead of killing the embryo, we can clone the embryo instead, and this perhaps will give us a better clue of how we can replicate stem cells. Now, we can clone embryos pretty easily. All we need to do is stuff the DNA from an embryo into another cell, and since embryonic stem cells are pretty much totipotent, they can grow into the entire organism. But let's say I'm George Lucas, and I want a bunch of extras for my next new Star Wars movie. Well, then you need to clone someone who no longer had embryonic stem cells. And so what you can instead do is take the diploid nuclei from the parent and then de-differentiate it by starving it, so then the DNA de-differentiates and becomes sort of like a stem cell. Then you can stick it inside a fertilized egg, and then you get a clone. And this is what happened to Dolly the sheep, which was a really big deal because we managed to clone an animal. However, Dolly died early, probably because they didn't differentiate the parent nuclei enough, and this is one of the complications that arises with animal cloning. Now, I mentioned pluripotent stem cells earlier, and it turns out we might not have to clone babies to make stem cells after all. Instead, we can use adult stem cells, like in bone marrow, and induce them to become pluripotent. These are called induced pluripotent cells, and they might be the future of medicine. For instance, we could take the adult stem cells from bone marrow, turn them into induced pluripotent stem cells, and then they could differentiate into a variety of different cells that we could then implement into medicines. And finally, what are some of the tangible uses of genetic engineering or DNA analysis? One of the big things, of course, is forensics, which, if you see TV shows, they very often use DNA analysis. For example, gel electrophoresis, analyzing R flips, and more recently, using short tandem repeats, which is basically repeating segments that actually turn out to be very efficient at recognizing different suspects. They're basically short sequences of DNA that are repeated over and over again in a DNA sequence, but the number of times that it's repeated may differ. For instance, like in this drawing, you can see that these two individuals have 3 and 4 STRs respectively, and by comparing this among different genes, you can accurately determine the suspects of a certain crime. Gene therapy is also a big thing. You basically just plug in genes from somewhere else into a person, and they make a thing that they can't make normally. Drug and gene product farming is basically you insert the gene into a farm animal instead, and the farm animal makes the thing that you want. For example, there's a thing called a farm goat where you stick the gene in them and they will produce the product in their milk, which you can harvest much more easily than if you had to just put in a bacteria. You can also use it for environmental cleanup, and another of the more controversial things is agriculture regarding genetically modified organisms, which of course is a topic that you should probably go to a political channel to watch instead. That was biotechnology.
And I hope that was helpful.
I'm loving it.